All right, let's get started. Again, we apologize for the delay. There were technical issues uh, on the ocean sciences side of things. So this is Town Hall 33, and the subject is accessing and utilizing the GeoTraces 2021 intermediate data product. I'm Bill Landing, and I'm joined by co-hosts Catherine Jondell, Rainer Schlitzer, Phoebe Lamb, Maite Maldonado, and Elena Mastrododas, who is going to run things for us. Next slide. All right, so the agenda is I will continue with this brief welcome. Catherine will introduce GeoTraces to those of you who are not super familiar with it. Rainer Schlitzer will talk about various ways of accessing the intermediate data product. Phoebe Lamb will then give a demonstration on working with IDP 2021 and WebODV. Maite Maldonado will talk about the links that have been added between trace elements and isotopes and genomics data. And then I'll finish up with a brief demonstration of how to register data for the next IDP using the DOOR portal. Next. Elena wanted me to emphasize that for those of you Twitter and Facebook users, you can tag your communications with these GeoTraces tags. So you're all muted by default. Use the chat to ask questions. Your video should be off for bandwidth purposes. You will not be able to share your screens with us. The closed captioning is enabled, and of course that covers up things at the bottom of each slide, which is unfortunate. The town hall will be recorded and available for 6 months, at least 6 months after the meeting at the Ocean Sciences 2022 website. And then all the presentations and the recordings will also be made available at the GeoTraces IPO site shown here. Next. All right, so I'm going to hand it off to Catherine Jondell, who's going to discuss GeoTraces. Go, Catherine.
Thank you, Bill. Hello, everyone. I am Catherine Jondell. I am working in Toulouse as a senior scientist of the International Project Office. So I will introduce you GeoTraces and why we decided to do an open source for our data. Please, next slide. So GeoTraces' mission is to identify the processes and quantify fluxes that control the distributions of key trace elements and isotopes in the ocean, and to establish the sensitivity of these distributions to changing environmental conditions. We are scientists from more than 35 nations, which are gathered and coordinated, and we are involved in the program, and our ambition is to study all major ocean basins and to establish the status of many trace elements and isotopes. Today, 131 cruises were completed, 43 full sections were completed. Almost 1,800 publications has been published, and they are in a database accessible from the website. And we published so far 3 open source data products, which have been released in 2014, 2017, and the last one, 2021, which is the one we will discuss today. Next slide. Just a rapid look at the map. The map represents the sections and the process study of GeoTraces. And here you have the section. The sections underlined in yellow or black are done, and the red one have to be done. So as you see, we already covered considerably the Atlantic Ocean, the Arctic Ocean, and the North Indian Ocean. We still have works to do in the southern part of the ocean and in the Pacific. Next slide. So the IDP 2021, by releasing, so the goal is very important. Why do we do this IDP operation? What do we open our data? by releasing and sharing more hydrographic and marine geochemical high-quality data, we wish to strengthen and intensify the collaboration within the marine geochemical community itself, but also to invite colleagues from other communities to join us. So the IDP 2021 consists on two big pieces: a compilation of digital trace metal data, so more than 100,000 samples from 77 cruises, and an atlas, eGeoTraces atlas. Here you have two representations. On the left, the map of the kind of station and sections that you can access to, where you will find a treasure of trace element and isotope data. And what is represented on the right is a 3D representation of the dissolved iron in the Atlantic Ocean, where you can see that all along the mid-oceanic ridge, there are input of iron, as well as along the margins. Next slide. Providing an open source atlas of data which are sensitive, contamination prone, it's a challenge. And for doing that, we have to go through exigent intercalibration procedure. So how does the SNI committee, we have a committee which is dedicated to validating our data. And so we have to go through this filter when we provide data. So the analyst is contacting the standard and intercalibration committee. Then he sent his report. He or she explained the critical aspects of the method. And the external calibration is depending on several criteria. We have to measure reference material, if possible, to analyze samples at the same station as another GeoTraces member, provide lab intercalibration exercise if we cannot do this crossover study, or any other external validations that justifies the quality of our data. And then, if they are validated, the data can enter the IDP. If not, we have to redo the exercise. Next slide. So then the IDP is done. How do you access to this IDP? So this is a screenshot of the website. And you can see the arrow allows you to target where the data are accessible. And then on the next slide, you, for example, can access to the eGeoTraces atlas. And then I will pass the word to my colleague, Rainer Schlitzer, who will explain you more about the atlas and about the database. Rainer.
Thank you, Catherine. Welcome to everybody. I'm Rainer Schlitzer from the Alfred Wegener Institute in Bremerhaven. The next few minutes, what I want to do is to show you how you can access the resources of the IDP 2021, which is the graphical atlas, but then also the actual data. So you go all these routes via the IPO, via the GeoTraces web page. You click on the data page, and then on the left side are all these links that you use for accessing the atlas, which we will do immediately now, but also the actual data. So eGeoTraces, the electronic atlas. So you click on this link here, and that takes you to, Elena, next slide. It takes you to the map on the left-hand side here. You operate this by filling these boxes on the left top, and you choose a group of parameters and then an actual parameter. So here in this case, we are having this dissolved trace elements, and we have chosen iron. Once you have chosen a group and a tracer, the map changes dynamically, and all the lines, all the sections that have data for the parameter that you have chosen, they turn red, including with their label. And all basins, ocean basins that have a 3D animation for this particular tracer that you have chosen, they have a blue rectangle active. Overall, eGeoTraces has almost 1,500 sections and almost 270 3D scenes. So it's a rich resource, many parameters along many lines in many basins. So if, for instance, we are interested in one of the sections here in the South Pacific, the label that is marked in green, you click on this label, and what you will get is the section of the tracer that you have chosen, ION in this case, along this line. And that is displayed in the right top of this slide. In addition to actually showing the graphics of this parameter, there's lots of information at the bottom of the page, including a description of the parameter, the list of names of people who have created the data, very important. So you always know who were these heroes making these data. But then also further links that take you to other tracers along the same section, but also eventually to these 3D scenes. And if you have clicked Pacific for these 3D scenes, you will see the example here shown in the slide bottom right. And actually on the website, this will be a rotating scene. You can look at the distributions from all angles. So this is basically the virtue of the eGeoTraces Atlas, providing lots of graphics in an, I would say, easily accessible way. And once you start, you have lots of links that take you further. And if you have lots of time, you can spend an afternoon analyzing what the GeoTraces community has actually achieved. Okay, enough for eGeoTraces. Next slide, please. So access to the actual data from IDP 2021, we have three ways, which we quickly will go through. The first one, bulk full package download. If you simply want to have all the data from the IDP 2021 and then use that, this is the way to go, first option. Very often, people don't want the full package. They have a very specific need. They want to have data for a specific domain, maybe time window. And instead of having all these 450 parameters, maybe you just are interested in 10, 12 or so. Then option 2, that's for you. You will be able to select what you want and just extract those data you want to work with. The third option actually will not require you to download any data, and also you will not be forced to install any software. You will do the analysis online, and this will be option number 2. I want to emphasize that unlike for previous IDPs where you had to register and you needed to log in with your specific name, now you have to agree to the Fair Data Use Statement, a document that is distributed with the IDP. Once you have agreed to it, you will get access. There will be no need for registration, no login needed. So the access is much simpler. So let's quickly go through these three options. Next slide, please. So the bulk download, the top link that you would choose, that takes you to a page at the British Oceanographic Data Center, which is our data assembly center for GeoTraces. You will see the page looking like this. There's a download link in the middle of the page. That will allow you to download the data of all these 5 packages that the IDP consists of. Seawater discrete sample data, this is the core of the IDP. But we also have sensor data from the CTD package. we have aerosol data, precipitation data, and cryosphere data. And you can get these data in 3 different formats: as ASCII CSV, column-oriented spreadsheet format, NetCDF if you are a modeler, or ODV collection for those people who use ODV for their analysis. So that will give you the full package. Next slide, please. Now, if you want to subset, then you would go for the second link, and that will take you to the Web ODV extractor, as we are calling it. And that will allow you to make a subsetting following 3 steps that are highlighted in this navigation bar near the top. And the first one allows you to select cruises, a geographical domain, or a time window. So here in this example, we have focused on the South Atlantic and have chosen just 3 cruises. This you do on this first page. Second page, next slide, please. That allows you to select the parameters or the variables for which you actually want data. And this is provided in a tree view here in the middle at the bottom, where you can select or unselect entire groups by clicking on this top layer box. Or you can select or unselect individual parameters. So in this case, we have made a quite limited selection. On the right side of this screen, you get a summary of what you have selected. So the number of stations, 118 out of, and the small number of variables that you have selected for output. Next, final step is number 3. Where you initiate the download, you choose a format on the left-hand side here, ASCII spreadsheet, ODV collection, or NetCDF. Once you click one of these buttons, a few seconds later, you have the package as a zip file on your computer. So that's the virtue of having this extractor. So you only get what you are interested in, much smaller packages. Next slide. So then there is this third possibility of having data exploration online. So no need for downloading the data on your private computer, no software download. And this online service, this will now be demonstrated live by Phoebe Lam, and I'm handing over to Phoebe.
Thank you, Rainer. I'm Phoebe Lam. I'm from the University of California, Santa Cruz, and I will have the privilege to demonstrate this online visualization tool. This is new for the IDP 2021. In fact, this tool is quite new, coming from Rainer. Rainer created all of this, so we're very glad to have him here. And now that we have the IDP inside this online visualization tool, it makes exploration of the data much easier and is an enormously helpful tool for teaching. So let me share my screen. Share screen. Let's see. Okay, so here I am at the GeoTraces webpage, geotraces.org, and I go to data, just like was shown before. And underneath the GeoTraces intermediate data product, I go to data exploration. You could also go directly to explore. I need to move, zoom around, explore.webodv.avi.de. And then you're here shown all of the data sets that are preloaded into this online tool. There's different data sets from different parts of the Earth system. Most of them are in the ocean, and there's so many useful ones. I use them, many of them, for teaching all the time. But for the purposes of IDP 2021, let's go to Tracer. And under Tracer, you see data sets from these different programs, including GeoTraces. So I'll click on GeoTraces, IDP 2021. And now you see these 5 different types of data that Rainer talked about. Cryosphere is brand new to IDP 2021. It has some snow and melt pond data. But I'm going to click on seawater, as that's probably what the majority of people are interested in. These are the data from the water column. And then I will click on the GeoTraces seawater discrete sample data set. So that will now launch this fair use data agreement. So as Reiner says, this is a new way of doing a data use acknowledgement that allows us to actually implement this type of usage. So please read this, and it helps guide users for acknowledging the hard work that everyone put in to create this data set. Once you've read it, then click I agree. And you're presented with the map showing all of the data in IDP 2021. So the new data, there's a lot of new data in the Indian Ocean. There's a lot of new data in the Arctic Ocean, new data in all ocean basins, really. If you're interested in the Arctic Ocean, this particular default view is a little hard to see. So I'm going to right click on the map, go to properties, click on projection, go to North Polar projection, click apply. Now we see a North Polar projection. We see the new Arctic data set that is in here, including the meeting of the Canadian, the US, the German GeoTraces programs in 2015 at the North Pole, or in the Arctic Ocean. Okay, so let's go back to the default projection, properties, default, apply. And what I really like about this, oh, and let me get back to the global map. What I really like about this is you don't need to download anything. You don't need to deal with software downloads. When I was using ODV for teaching, I had to get the computer lab and have people download the software, the students that is, download the software, have them load the data sets, and there were always complications. Now, this year, I managed to do this all online. The students had no problems at all accessing this. I love this. So for teaching, another thing that's really great to do is to show a meridional section, let's say through the Atlantic, then you can show water masses. So let's do that. I'm going to define a section. So I right click on the map, I go to manage section, define section. And now you use your mouse to click a line through which you will, that goes through the data points that are on the map. And I'm going to be following, for those of you who know GeoTraces, this is more or less the GA02 line that the Dutch ran. And it's a beautiful meridional section that shows so many wonderful things that one uses for teaching. So I will use this again. So now I've defined a nice meridional section. I will make latitude as my section coordinate. So that will show on the x-axis. And maybe I'll give it a name, Atlantic Meridional, let's say. That way I can, whoops. That way I can come back to it later. I'll click apply. Okay, so now I've defined my section, but I want to see plots. How do I do that? So I right click here in this white part and go to layout, go out to layout templates, and you see these different options. So ODV has these 4 different types of plots: station plots, scatter plots, section plots, surface plots. Today, I'm going to show you how to make some section plots and scatter plots. I tend to like to start with 5 section windows because it creates them nicely evenly. So let's start there and then we'll modify it from there. So these are the default axes that ODV will generate for you. And you have always the section coordinate on the x-axis. So we have latitude here, and then pressure on the y-axis. The Z's, the color bars, the Z's are not particularly useful here, so let's change them. And what I'd like to do in my demonstration, and what I did for my class, is to show that the major nutrients show water masses moving through deep water formation, and the micronutrients follow some of the major nutrients. So that's what we're going to demonstrate today. So I'll right click on this top left map, I'll go to properties, and let's plot phosphate. So we'll go to data. We see it's a section plot. We've got the right X and Y axes, but not the right Z axis. So I'm going to go to 37, which is phosphate, and I'll click apply. And this is now the phosphate data taken along wherever these data points are from. I'm going to make this, I'm going to interpolate this to make this a little prettier. So I'll click on, so I right clicked, I click on display style. I'm going to grid it. That will interpolate the data. I'll keep these automatic scale lengths. I personally like to make these marker sizes a little bit bigger so I could see where their data were coming from. I'm going to apply to all windows because that way I won't have to redo these property things for each window, and I'll click apply. Okay, so now we have this nicely interpolated data set showing phosphate. We see the North Atlantic deep water has a lower preformed phosphate concentration than Antarctic bottom water, which is super high. We see Antarctic intermediate water. This is great. So now let's add another major nutrient. Let's add silicate. So I right-clicked up here in this top right. I'm going to go to data. I will click on silicate, which is 39. 39. I will click apply. And we see it's a little bit different, much lower silicate in the North Atlantic, very high in the Antarctic bottom water, but much lower in the Antarctic intermediate. Now this is geotraces. This is not just hydrography. So we are interested in trace metals. And so now I'm going to add cadmium. And we're going to see how cadmium relates to these major nutrients. Apply. Okay, that's interesting. I'm going to add zinc, another important micronutrient. Go to zinc, that's 110. Down here, click apply. And look at this. This is what I have my students plot. And I asked them to tell me what cadmium looks like. What does zinc look like? And visually, you can see right away that cadmium looks like phosphate and zinc looks like silicate. But let's say you want to actually show that, make a scatter plot. So we'll make now 2 scatter plots. I don't need this 5th section window, so I'm going to make more space. And I'm going to zoom in here. And also the way ODV plots scatter plots is it plots whatever is in the map window. So I'm just going to focus on the Atlantic. And I zoomed in just to the Atlantic. That also restricts the data that will be plotted, but also gives me more space. I will now right click on this last section plot that I'm not going to plot a 5th section. So I'll go here and I'll click on properties. I'll make this into a scatter plot. I'll make this into, I want X on my X axis, I want phosphate. Oh, you see here, there's 584 parameters. Lots of parameters in IDP 2021, but we want 37 because that's where phosphate is. Phosphate. And on the Y axis, I want cadmium because I want to see how cadmium relates to phosphate. And that is 85 over here. I'm going to keep as my z-axis depth, so that will show me in colors what depth the plots, dots are from. And now we have this nice scatter plot that shows cadmium versus phosphate. Okay, that looks like a pretty good correlation. There's a little bit of a kink, and of course, there's a lot of oceanography to talk about there. But what I'd like to do is now, whoops, that's not what I want to do. I want to lay out. I'm going to make that a little bit smaller there. I'm going to move it over and I'm going to make another one. And I want to see how zinc is related to phosphate because we know zinc is a micronutrient. It's used for all sorts of enzymes. So let's create a new window and I will right click and move that. Here we go. And I will go and plot zinc on my y-axis. Zinc is down here. And so we know it's a micronutrient. Shouldn't it correlate with phosphate? Not so good. So I'm just talking you through a problem set that I have my students do. Okay, and so now we're gonna plot silicate on my x-axis. And silicate is here. And now we see a much better correlation. So this is just, I've taken you through half of a problem set, homework 3, I think. And it's just a wonderful tool, both for preparing slides for teaching, which I use all the time, also for teaching students. No one has to download anything. One thing I did want to say is you can save your view here. So let's say I want to say IDP 2021 test. That means I can come back to it later. As long as you don't clear your browser history, you can come back to this later and play around as much as you can. One other thing that IDP 2021 does is that for the first time, we've linked to genomics data. And so I will now hand this off to Maite Maldonado, who will tell us about that.
Thank you so much, Phoebe, for that. So I'm going to take over here and I'm going to share my screen. Okay, so I'm Maite Maldonado at UBC in Canada, and I'm going to share with you how we link omic data to the wealth of TI data that we have in the IDP 2021. So as a community, GeoTraces really wishes to strengthen and intensify the collaboration between the marine geochemical community and the biologist omic community. So now one of the goals of the IDP 2021 was to provide these linkages between these TEI data and the omic data, as there were multiple cruises where omic samples were collected by very nice geochemists for a number of labs. So in this map, what we have is the cruises where these samples were collected. So there are 3 cruises in the Atlantic, and 1 cruise in the Pacific. And what you see here, these little dots, is where we have single cell genomic data, and the gray symbols are stations where metagenomic samples were collected, as well as 16S and 18S. So this is one of the goals that we had. And just to tell you what data we're trying to link, we have linking to the IDP 21, 480 metagenomes in the ocean, 14 single cell genomes, 273 16S, 18S RNA gene analysis. And we also have 480 microbial communities, taxonomic and functional structure and abundance. So that's what we have. But one of the major challenges we faced when we were trying to do this is that the omic data is dynamic. Changes occur depending on the ever-growing genome reference databases, while the TEI data is static. So the solution we came up with is to provide linkages to the linkages in the IDP 2021 to the omic data. I want to really acknowledge here Rainer Schittler for making this happen, as well as Paul Berube and Jesse McNichol. Without them, this will not have been possible. And I also would like to thank anybody and everybody that provided omics data, and especially members of the Penny Chisholm Lab. So what we came up with is 4 types of sample descriptors for the omic data linkages in IDP. So in essence, we are going to take you to the NCBI website, which is a bioinformatic site, where you can find the metagenome, the single cell genomes, and the 18S and 16S rRNA. And we are also going to take you to the EMBL EBI website, where you can actually visualize some analysis of the data already. So the data descriptors that define these omic data linkages were implemented into the IDP as metadata variables. So I'm going to show you in a minute, but we are going to go and look at metadata variable 11 and 13. These are the accession numbers to the metagenome and the 16S, 18S rRNA in the NCBI website. Metadata variable 12 is going to give you the accession number for the NCBI single cell genome. And variable number 14 is a place where you can click and go directly to the EMBL EBI magnify analysis. What's very important is that all these, the link between the omic data and the GeoTraces data is really the bottle number. So everything is tracked by a bottle number, and that's how you link your data to the chemistry data. So I'm going to take you now to the same place where, so I'm not going to repeat what Phoebe did, but in essence, you are going to get to this. This was one of the first clicks of Phoebe. So here we have a summary of all the available IDP data. And what I'm going to do, imagine I'm an omics scientist and I want to look at what data is here. So what you are going to do, you are going to click on the right-hand side of the mouse and you are going to go to a station filter. So I want to look for stations that have omic data. So you're going to go to this tab that says availability. You're going to click on it. And I'm going to look for my omic data, which is, remember, variable 11, 12, and 14. So I'm going to click and I'm going to hold Control key. And you're going to select these 4. These are the omics data. And I'm going to say, I want to look for any station that contain any of these type of omic data. So you are going to press the OR. And you're going to say apply. This is going to be processed. And these are all the stations that have one of those 4 at least omic type of data. So you can see the red dot here is in station 2544. And I'm just going to move it to, I want to go to station 270. I want to go here. I want to go there, okay? So what you see here is on the top, you see the cruise name, you see the station name, longitude, and so on. And then here in the bottom, you see sample 1, 48. So this means that there were 48 samples collected in these stations and you're looking at sample 1. You can see that this sample was collected at a depth of 8 meters. And this is this key number that I'm telling you about, BODC bottle number. This is linking genomic to chemical data. Now, so it looks like sample 1 actually has genomic data. You can see how there is a little number here in variable 11, another one in variable 12, and 14, nothing for 13. Ooh, this is a sample with omic data. What else is there? So you click on this little arrow, sample 2 at 11 meters, nothing. Sample 3 at 25, nothing. So, ooh, sample 5, also there is genomic data. I have 11 and 14. And you can explore the data like that. Ooh, 7, again, depth 74, I have more omic data, okay? So you go here and what you're going to do, I'm going to do this in a second, but let's get this number right here and we are going to go to the NCBI website. So I'm going to go to the NCBI website and I enter the number, okay? You're going to click search. And that's going to bring you to the metagenome data. And you, for the omics people, you know how to work this website. There's a lot of things you can do here. And again, I just want to emphasize how the bottle ID here, it's what links this data to the GeoTraces data, okay? Let's go back. I'm not going to go to, I want to go back. I'm not going to go to the single genome data. Let's go here. I'm going to go to this one. So this is a website that you can click. And this is bringing us to the EBI website. And in essence, they have analyzed the metagenome data in variable 11 by taxonomy and functionality. So you can click on here. And what we have here is we have a taxonomic analysis, but for the small subunit of RNA. And what we get here is the community composition of that sample. And then, you know, you might love Flavobacteria, so you might want to click on them and see who is there or whatever. And these are different tools that you can use. So you can tell me that that sample has 88% bacteria and 7% eukaryotes. So that's for you to play, but you can do both taxonomic analysis and functional analysis. So the last thing I want to show you is how to get this data. So the first thing I want you to do is if you go to the Browns info file, you actually are going to get those websites that we were talking about where you can go with your accession number. So here is the NCBI website that you want to go to and so on. So it's all here in the information in the info file. Now, let's say you really want this data. This is great. I'm so excited. So you go to data and you say, okay, I want an ODB spreadsheet file. So you click on it. And now I'm going to say omics GA02 station 16B. And you are going to say, okay, that's the name of my data set. So here, the first thing you have to click is the metadata variables. These are things like cruise name, chief scientist, and so on. So you can just select all. or some, let's say we select all. And then you get to the variables that you want. So this is where I'm going to say, I want depth, and I hold in your control key, I want BOD bottle number, and I want all these wonderful ohmic session numbers, but I also want iron. I want to study iron and copper with this data. So you're going to click, and you're going to get the iron and copper data associated. So here is iron, and here is copper and iron. So this is the associated data at that station. So you're going to select, and you're going to start your export, and a file is going to be created for you. I'm just going to click here. And this is how your file looks like. And then you can import it into MATLAB or Excel. So here are your accession numbers for those websites. And if you scroll here, these are the numbers for your iron and no copper. So this is an amazing tool, and I hope omics people are excited to explore the geochemical data and vice versa. So please come and play. And now I would like to hand it over to Bill Landing, who is going to tell you about how to join the DOOR portal in order to include data in the next IDP.
All right. Elena, if you can launch the other presentation, the DOOR portal presentation. Oh, Maite, you have to stop sharing. There we go. All right, we're running a little bit late, and I don't know if we'll actually go through all of these slides, but I do want to start with an introduction, and that is you might have data that you would like to contribute to the next GeoTraces intermediate data product, and that would be, of course, data from cruises which are occurred after 2020 and cruises that are occurring up until we produce the next intermediate data product. So the way you do that is you, as always, you go to the IPO website, geotraces.org. Next slide. There's two ways to get into the DOOR portal. You can either hover over data and go to the GeoTraces DOOR, or there's actually a direct link to the DOOR portal. Next. And the DOOR portal is the Data for Oceanic Research portal. This is a place to register the data sets you want to have included in the next IDP. Next. So this is not where you submit your data. This is where you register your data. But the data has to be submitted to the relevant US, Dutch, French, or Chinese National Data Center, or directly to the GeoTraces Data Assembly Center at BODC. And you will get there's a data submission template which is available through the DOOR portal, and you can use that. Next. So the software, this is an incredible tool. It generates a database of all the scientists, the cruises, and the parameters so that we can track the entire process of registering data for the next IDP. Next. There are tutorials, a PDF tutorial, which is very detailed, and a video tutorial that Phoebe put together. These are highly recommended when you want to start getting into the submitting your data for registering your data for the next IDP. Next. We use ORCIDs. So you have to use your ORCID to log in. If you don't have one, you'll be prompted to register for one. Next. Every time you log in, it's going to ask you to confirm your email address and your affiliation so we can keep track of people, because people move around and we want to make sure that we maintain an accurate list of the users. Okay, next. So it's already been discussed, but we've eliminated the registration step to look at the IDP data, and we've switched to this fair use agreement. You should read the fair use agreement, understand its intentions and applications. And again, the goal is to recognize the incredible hard work that all the scientists put together to generate this data. Next. So that will take you into the actual portal, where there are actually 7 steps here, 8 steps. The first step is to register your data sets. Next. So a data set is defined on a cruise by cruise basis and a parameter by parameter basis, because that's how the intercalibration process is organized. Next. So a couple of questions. You may be working with colleagues. If a data set was generated by more than one scientist, each scientist can register their portion of the data set, or one of the scientists can assume the responsibility for registering the entire data set on behalf of the other scientists. If you have any questions about any of this, you can contact Mohammed Aju at the GeoTraces Data Assembly Center. His email is shown here. Next. All right, there's many steps in registering a data set and too many to cover. But if you watch the video tutorial, read the PDF tutorial, each step is explained in great detail. The screenshots are shown. But you can register data from a GeoTraces cruise, a section cruise, a process cruise. You might have data which you think is compliant with GeoTraces. And so perhaps you have dissolved trace element data from a cruise that you completed. And you want to know if that data is appropriate to submit into this into the GeoTraces data product. So you can select your cruise and you can select your parameters from the extensive lists which are buried inside step 1. Next. Select a cruise, you just scroll down. You can search for the cruise name. If you put in GAO2, you'd see that Atlantic section that people have been discussing already today. Next. If you have compliant data, again, this may apply to many of you, click that button and there's more details at these 2 websites where you can understand what compliant data actually means from the GeoTraces perspective. Next. There's 2 ways to find your parameters. There's a search tool and a tree exploration tool. You can use either method. You can use both methods. When you've identified a parameter, for example, it's showing here, it's hard to see, but an aerosol acetate parameter has been selected. If I hit the plus button there, it would add it to my list. Next. And when you're done, you say OK, and you'll get this pop-up. Next. So here I've selected all these parameters that I'd like to register. I obviously must have done a vertical profile, and I've got a bunch of dissolved trace element data, and I want to confirm that this is the data set I want to register. Next. All right, I'm going to skip step 2 and jump to step 3 really quickly, and that is the critical component of GeoTraces is ensuring the accuracy, reliability, and intercomparability of the data. This is an essential component of GeoTraces. You have to generate intercalibration reports, and those will be reviewed by the Standards and Intercalibration Committee. Next. So that this S&I works with the PIs. They will work with you to make sure that your data is the highest possible quality. Next. So there's instructions on how to create these reports. Next. If you select a cruise where you've registered data sets, so KN204, that was a US GAO3 cruise, I believe. And here's all the parameters that I had selected. And if you'll notice, next to each parameter, there's a 6-digit barcode that has been assigned automatically to a unique 6-digit identifier. Iron data from that cruise submitted by myself and my coworkers from my lab. That data will be tracked using that 6-digit barcode. That will allow us to follow the data process from the submission to review to finally appearance in the next IDP. Next. It's essential that you don't change these headers and barcodes because this is how we keep track of the data. Next. You can download an intercalibration report template for each parameter that's shown here. Are these little blue Word files. You can download a template. Next. Then you can upload that template. Once you've completed that report, you can upload it back in step 4. And you should use the intercalibration report templates that you just downloaded, edit them, submit them, and the Standards and Intercalibration Committee will jump on it and start communicating with you, again, to make sure that your data is of the highest possible quality. Next. Keep going. Next. Yeah, so here's, okay. You can also download data submission templates. These files will show your data set registrations and every parameter with its barcode. And if you haven't already submitted your data to your data center or to the GDAC, it would really be good to use this file to structure your data. Next. I just said that. Next. All right, so this is what it looks like. It has some metadata in these orange colored cells that we prefer you not edit. So it shows the your name, your ORC ID, the cruise ID, the GeoTraces cruise ID. Next. Oh yeah, don't pay attention. We don't want you to edit certain cells. Next. All right, we recommend that you use the CDataNet quality flag scheme, and that's explained if you go to that link. Next. And then here you need to enter information that's consistent with your cruise logs and cruise reports. For example, there might be an event ID number for each station. There might be a sample ID number, sample depth, coordinates, date and time. These are all things that would uniquely identify your data set. So you need to enter enough information that, that your data set is clearly unique and different from someone else's. Next. This is the key part. Now, if you look in what's in the little red box now, you see dissolved iron concentration from a bottle, and it has a little, that 6-digit barcode, DU1, I can't read it. But the point is, don't edit those. This is how we keep track of the data. You could enter your data now into this spreadsheet. And it asks you to enter the actual data in the preferred units, the standard deviation, and the flag code for each parameter. Next. If you use this, then you could submit it to your data center. Next. If in many cases, you might have already submitted your data to your national data center, and you wouldn't have used this format if you didn't go in through the door portal. It's really helpful to put together the next data product if everyone uses templates like this. And you have to communicate with Mohammed at GDAC to discuss the most efficient way to get your actual data associated with these data set registrations from the door. It's actually easy to do. You may have to actually just reformat your data, dump it into this template, and resubmit it. And the data centers are fully aware of this and are helpful in getting it done. Next. All right. If you go back to step 2, we want to make sure that the data, the PIs, the data generators, the grad students, the postdocs, and analysts, those people who are responsible for a given data set, get associated with that data set. And we do that, again, using ORC IDs. You can return to step 2 at any time to complete or modify this information. Next. So you just need the ORC IDs and institutional email addresses of the people who are responsible for the data set. Next. Step 5 is you have to tell us that it's okay to use your data in an IDP. So you can select cruise by cruise, parameter by parameter, to allow us to use the data in the next intermediate data product. And you can change your mind if you like. Next. In step 6, if your data has been published, if there's a DOI for the paper or a DOI for the data set, please enter that here, because that way we can make sure your data are correctly cited. And if the publications are not yet available, then you can always return to this page when they're published and link them to your data. Next. The last step, list my data sets. You can follow this whole process with every data set you've registered from the List My Data Sets link. Next. For example, you can see which scientists have been linked to each data set and the principal investigators. Next. You can see when the S&I committee has received and approved your intercalibration report. Next. All right, so again, there's much more information on the tutorials for using the door portal, and I encourage you to look at that, and you can, of course, contact me at any time. But what's really important is to thank everyone who contributed to the IDP 2021, and that is, of course, the people who generated the data, the the members of the Standards and Intercalibration Committee. They work very hard. Data Management Committee, the Parameter Definition Committee, of course, Mohammed and his team at GDAC, at the BODC, they are responsible for processing all this data and handing it off to Reiner. The DOOR portal, the software was actually developed at Toulouse by this team. It's a fantastic development for us. WebODV, again, you've already heard from Reiner and his team. Catherine and Elena at the International Project Office, and then the people at the National Data Centers who have just helped us immensely in getting the data properly formatted and transmitted to get into the data product. Next. And of course, it's important to acknowledge the financial support for SCORE. This is supported in large part by the U.S. National Science Foundation through a series of grants and contributions from other member countries in GeoTraces. Next. So if you have any questions, make sure, ask Elena first. She has more answers than I do, but then you could ask me. And I think that's it. So at this point, I'm looking to see if we have any chat questions. All right, we've run long, and I think we're done. Is there, if there's any other chat questions, we're going to record, the chat will be recorded. The entire presentation has been recorded. That will be available on the GeoTraces IPO website. I'd like to thank all of our speakers today, with Catherine, Reiner, Phoebe, and Maite. I guess we're done. Does anybody else have anything they want to add?
Use the IDP.